Good afternoon. Namaste. This is Dr. Uma Keduvittan, Faculty of Agricultural and Veterinary Sciences, Jayoti Vidyapeeth Women's University, Jaipur. As you know, I'll be your instructor for Metabolism and Bioenergetics course this semester. And we have been looking into the biosynthesis of essential amino acids. So in the previous session, we have looked into the biosynthesis of histidine and so we have looked into the formation of the precursors for purine synthesis and from phosphoryl ribonucleotide pyrophosphate and ATP, how the synthesis begins and how histidine can be formed in a multi-step reaction in bacteria and plants that have been looked at in the previous session. So in this session, we will be looking at the biosynthesis of 6 amino acids. So we have threonine, methionine. So we have lysine, methionine, and threonine, and isoleucine, valine, and leucine. So the sum total biosynthesis we will be looking into in detail. So over here, actually, we will be looking at each of these steps in a very step-by-step fashion only. We will be looking into all these pathways, but they are actually all interconnected, and that is why this has been expressed as such a huge pathway. But we'll be looking into each of them individually. So first, let us look into the biosynthesis of lysine. So you know that lysine, methionine, threonine, isoleucine, valine, and leucine, they are all essential amino acids. So our 9 essential amino acids are this lysine, methionine, threonine, leucine, isoleucine, valine, and then what we have studied: histidine, phenylalanine, and tryptophan. Tyrosine is not considered to be an essential amino acid, but it is conditionally essential amino acid if enough phenylalanine is not supplied, because phenylalanine hydroxylase catalyzes the conversion of phenylalanine into tyrosine in our body. So mammals have actually lost the ability to produce all these amino acids, because anything more than 7 step, we have actually reached a stage where they can be lost, because they can be obtained from the food sources that we are having, and so then it need not be synthesized in our body, and that is how evolutionarily, in order to conserve the energy, we have lost the ability to synthesize these multi-step amino acids that have like more than 7 steps in their synthesis from the primary metabolites that are available in the cell. So the, today we will be looking into the biosynthesis of lysine. So let us start with that. Yeah. So I will share the biosynthesis of lysine. So this first part is the biosynthesis of lysine. It starts with aspartate, and the enzyme that is involved in the biosynthesis of so the enzyme that is involved over here is so now aspartate will be phosphorylated by aspartokinase, and this aspartokinase will use one ATP and convert it to ADP and this phosphate. So it's an energy intensive process. And this phosphate will be attached over here. So it is an aspartyl beta phosphate that will be formed. And then the aspartate beta semi-aldehyde dehydrogenase. So this is a dehydrogenase enzyme that converts one NADPH to NADP plus, and that will actually remove this phosphate group as well and convert it to its semi-aldehyde form. So now we have a CHO over here. And once we have this, instead of the OP, now we have a H over here. And so that is the semi-aldehyde form. And once we have the semi-aldehyde, now from here, this could be used for the threonine biosynthesis as well. So this can lead to the formation of homoserine, and that is a metabolite in the formation of methionine and threonine. But over here, since we are first looking at the lysine metabolism, lysine anabolism, so let us look into these steps that are involved in the aspartate conversion of aspartate beta semi-aldehyde into lysine. So the next step is the formation of dihydropicolinate. So that is catalyzed by the enzyme dihydropicolinate synthase. So pyruvate is added over here, and once pyruvate is added, then one, excuse me. Yeah. So now that we have this, pyruvate will be added to this and dihydropicolinate will be synthesized. So one step is the addition of pyruvate, and the next step is the removal of water. And then what we will get is a ring structure, and that is the dihydropicolinate. And once we have the dihydropicolinate, then the piperidine 2,6-dicarboxylate dehydrogenase. So again, there's a NADPH plus H plus giving NADP plus reaction, and that will, this ring structure will form piperidine 2,6-dicarboxylate. So this ring structure is essential in lysine biosynthesis, and this doesn't happen in any of the other biosynthesis pathways. So this dihydropicolinate to piperidine dicarboxylate reaction, and then the addition of succinate, the succinyl CoA and water is added by succinyl 2-amino 6-ketopymalate synthase. So that is the next product also, N-succinyl 2-amino 6-keto-1-pymalate. So 2-amino 6-ketopymalate. So 6-keto-1-pymalate. So this will be formed when this ring structure is opened and there is a succinate added to this NH. So there is a double bond O over here instead of, and the COO and a double bond O, and there is an NH and the succinate that has been added to this. The coenzyme A will be removed. And then the next one is a transaminase reaction, where instead of this double bond O, we will have the amino group added. So that will be succinyl diaminopymalate aminotransferase. So once that happens, this succinate is not actually required. And but for this amino reaction to happen, this transaminase reaction to happen, we need this compound. So once, or rather for this ring structure to open, this compound is required. So once the ring structure is opened and the double bond O is formed, then we can do the transamination reaction and then remove the succinate by a desuccinylase enzyme. So once the succinate has been removed, then we will have alpha diaminopymalate. So that is the precursor for lysine formation. And then it will be epimerized to form, instead of the this one, the meso form will be formed. So the meso alpha epsilon diaminopymalate will be formed. So over here, we have the COO, the NH3 plus and the H are there. And then there are the two CH2. And then in here, only this epimerase, the chiral form is changed. So instead of NH3 over here and H over here, this will be removed because only to this meso form lysine can be formed. And so once we have this, then this has become the ring structure and this is a epimer now, no? And once we have that, then pyridoxal phosphate will catalyze the reaction of diaminopymalate decarboxylase. So lysine is an amino acid that is formed by a decarboxylation reaction. Like we have looked earlier into the decarboxylation reactions. So what happens is this CO2 is removed. This end CO2 is removed. So we will have CH2 over here, NH3, and then the 3CH3. And then this is the basic structure for any amino acid. So the COO, NH3 and H, and this is the R group for the lysine. So this decarboxylation reaction will lead to the formation of lysine. So that is the biosynthesis of lysine. Now, if we were to look into the biosynthesis of threonine, then we will have to start from the aspartate semialdehyde. So the aspartate semialdehyde can undergo a dehydrogenase reaction. So that is done by homoserine dehydrogenase. And once homoserine dehydrogenase adds the hydrogen to aspartate beta semialdehyde. So instead of now the CHO, we have the CH2OH, that is the homoserine. And once we have the homoserine, then threonine synthase can add the, no, I'm sorry, homoserine kinase will add a phosphate to homoserine to make it phosphohomoserine. And then threonine synthase in another pyridoxal phosphate catalyzed or in a reaction where pyridoxal phosphate acts as the cofactor, then we will add a water and remove the phosphate in order to form threonine. So this threonine is then involved in the serine formation and all. So like we will be looking at it in the next session what for the methionine biosynthesis and isoleucine, valine, and leucine biosynthesis as well. So that will be it for this session. Thank you.